Okay, we're going to start our unit on DNA and biotechnology. As I'm going through, you want to be taking notes. Feel free to stop and start again. Go back through any information you want to hear again. But make sure you're writing down and jotting down notes so that you can have it for later on. Okay? So we know our unit question is, what has a larger impact on my life, my genes, or my experiences? One of the quotes I've always seen, and it's always struck me, is a quote from Irving Berlin. And he said, life is 10% what you make and 90% how you take it. And it really made me think, am I really only in charge of 10% of my life? Is the rest predetermined? You can kind of think of this in context of our unit question. What is more impactful, our genes, our genetic information we inherit from our parents that makes you you, or do you control what happens? I guess to really understand that, we need to know something about genes. And to do that, we really need to know something about DNA. I'll bet if I stopped and asked people on the street, what is DNA? They would probably have some idea that it stores our genetic information and it's part of all living things. But just 60 years ago, the scientific community and world at large had no idea how important DNA was and what role it would play. If you look at this picture, it's a natural question to wonder, what is this made out of when you're observing anything? This is from the Cedar Forest in Glacier National Park, and it's not hard to wonder how these trees came to be. Why do they smell so good? Why are they so tall? What is bark? I mean, what is it? Why are trees different than humans? And how are they different from humans? We're both living. Or to observe the world around you and wonder at some point, what is this made of? To wonder what is actually in our food. What are the ingredients for a butterfly's wing or for a kitten? Humans have always been asking this question: who made us? How? Out of what? I think one of the most biggest questions of life is for a mother to wonder, how is this life growing in her belly, technically for 9 months, and well, I guess technically in her uterus for 9 months, and suddenly pop out a human being and watch it grow up? How does that happen? What are we? Possibly an even more prolific question has always been related to life and death and human health. How do we combat sickness? Trying to understand processes that we can't necessarily see. It used to be if you became ill, it was seen as a punishment for something you did. It was revolutionary to discover a microbiotic world of organisms, things we couldn't see with our naked eye but have a huge impact on our health. What about diseases that are passed on from one generation to the next? How does that happen? The key word is cells. Cells are the building blocks of life. Um, just like Alhambra is built made of bricks, all life, even bacteria, is made of cells. You've probably had some exposure to cells. You may even know the difference between prokaryotic and eukaryotic cells. You might know that eukaryotic cells have a nucleus and have a more complex system of organelles. And you might know that the current theory is that life began as prokaryotic cells and then formed symbiotic, interdependent relationships and became the first eukaryotes. You might know the difference between animal and plant cells. And you might know, oh, I know a plant cell because it has plastids, those big green chloroplasts, big green parts you see on the diagram that have the chloroplasts, and they carry out photosynthesis. And they have chromoplasts that color flowers and fruits. And they have vacuoles and strong cell walls. I mean, there's tons we could talk about here, but that's not really what we're focusing on. Different kinds of cells, but the key is understanding that life as we know it comes about through continuous cell division. That's how a baby is formed. Chromosome pairs separate, replicate, and realign. So the question is, where are the chromosomes? Where's all the genetic information located? For eukaryotes, the command center is the nucleus, which contains the DNA. What we want to focus on is the DNA, the genetic material that's stored in the cell and gives directions for making new cells. Why do we even need new cells? Well, it's for growth. Initially, it's for growth. How do you get from being little to big? Well, we have to make new cells. We can make new cells to repair damaged cells, maybe from sunburns, or cells that are always growing, hair and nails, never stop, or to replace the cells that are dead. When you stop replacing these cells as fast, well, especially in your skin area, in your fatty tissues, you get wrinkles because you're not replacing as many cells, and so you don't have enough material to fill out your skin and you start getting wrinkles. Another important function of the cells to give directions for making the body work. If the directions are incorrect, then your body doesn't function correctly, and that's the basis for inherited diseases. So I like looking at this diagram in the opposite direction. I see the twisted section of the gene on the DNA strand, and it is packaged inside the chromosome, which contains all the information, the blueprint, and the owner's manual for your body. This information tells your body how to make new cells and carry out cellular function. Cancer cells are cells that have DNA with an incorrect message, and the cells replicate uncontrollably, and a lot of times this can lead to a tumor. So how did this all get started? How did we come up with the structure for DNA and understand that DNA controls as a blueprint, it has the genetic information for your body? You can go all the way back and talk about Gregor Mendel and his pea plants and understanding hereditary traits and Louis Pasteur and vaccines. I mean, science builds on itself, but we'll try to limit it to a couple of key people. So in the early 1900s and late 1800s, pneumonia was killing people left and right, I suppose, and there had been some discoveries in vaccines. And so several scientists were studying pneumococcal virus. And one scientist, Frederick Griffith, he did an experiment with mice, which was pretty important. He was studying two strains of the pneumococcal virus. One is the R strain, the rough strain, and it's harmless. If it's injected to a mouse, it lives, it's fine. The S strain is the smooth strain, and it's fatal. If it's injected to the mouse, poor little mousey dies. So Griffith said, what happens if I heat this harmful strain? So he heats it up, and it's no longer harmful. He injects it into the mouse, and the mouse is healthy, no problem. But if he mixed the strain that was heated, so he mixed the harmful strain that's been heated with unharmful strain and injects the mouse, it dies, and that's not what he was expecting. And so Griffith decided that something is causing a change. There's a transforming substance, but he wasn't sure what that was. Is it DNA or is it protein in charge here? Well, that's where an American biologist came in, Oswald Avery, and he took the experiments one step further. And he thought that protein was the transforming factor here. So he took the heated treated deadly strain and the live harmless strain, and he mixed it with protein destroying enzymes. And the bacteria colonies were grown from the mixture, and they still transformed. So even though he had something in there that should destroy the proteins, it was still going to be harmful to the mouse. So he decided, maybe it's not proteins that's in charge, maybe it's DNA. So he did the mixture again, and he had DNA destroying enzymes, and this time the colonies failed to transform. So Avery concluded that DNA is the genetic material of the cell. Who's next? Rosalind Franklin. Okay, so there was a group of scientists working in Cambridge and in England in the 1950s. One of the important ones, Rosalind Franklin. You know that atoms are too small to be seen by a microscope. Why is that? Well, it has to do with light waves and their length. Anything smaller than the wavelength of visible light, you can't see it. So you need a smaller wavelength. You need something like X-rays, has a smaller wavelength. So Rosalind Franklin took one of the first images of DNA, and you see it there. All right. In the process, unfortunately, she was exposed to a lot of radiation, and she developed ovarian cancer, and she died from that cancer. But her colleagues, probably some of the most famous scientists that you might know, Watson and Crick, used her photographs to help them in their discovery of the structure of DNA. So, Watson and Crick knew that the structure of DNA had to consist of two chains. They used a lot of math to figure it out. And then they used a ball and stick model to come up with the structure of DNA. And what they figured out is DNA is made up of nucleotides. Nucleotides have three parts: a phosphate group, a sugar group, and a base group. There's four different types of bases: adenine, thymine, guanine, and cytosine. And they published a paper on their results, and it was a pretty short paper, but it was revolutionary. What happened to them? Watson went on to launch the Human Genome Project, and Crick went on to focus on nucleotides and biotechnology, like advances in genetic fingerprinting and screening for inherited diseases. They were both very successful. but their model is extremely important. So, what do we need to know? A helpful series of analogies. Maybe this will help. So if you think about, most cells possess a nucleus, which is like a big spherical library. I like to imagine that all the books in the library are cookbooks. It's just a huge library of cookbooks. So in this analogy, the cookbooks are the chromosomes, which are inside the nucleus. Okay, so keeping with the book, the chromosome is the book. Each chromosome is a different book and it's a different story, and the story is DNA. How many chromosomes do humans have? Humans have 46 chromosomes. We have 22 pairs of autosomal chromosomes, non-sex chromosomes, that can be directly aligned by shape and size. And we also have a pair of gonosomal chromosomes. Those are the sex chromosomes. So men have one large X chromosome and a smaller Y chromosome, and women have two X chromosomes. Other species have different numbers. The numbers of chromosomes don't tell you about its complexity. So there's a fern that has 1,000 chromosomes, even though humans only have 46. It's just kind of the way things are. The chromosomes are ordered by numbers. So for humans, we have 2 chromosomes number 1, 2 chromosomes number 2. One is from your mom, one is from your dad. Some organisms are haploids. They only have one set of chromosomes, like bees and wasps. Others are octoploids. We know about strawberries, and some even have 16 sets of chromosomes. Human sex cells, the male sperm and female egg, they're haploids. So when they join up together, we keep the same number of chromosomes. So we don't just keep getting babies that have more and more chromosomes. One set from mom, one set of dad, and end up with 46 altogether. So it's all about combinations and interactions. If you Googled a recipe for chocolate cake, you could find hundreds of options. Well, what's the difference between one and the other? Well, if you know about baking, you know that adding salt or not adding salt can really affect the taste. You know that maybe warm water and sugar activate yeast. It's got to be warm water. You know, different consistency comes when you use honey or applesauce instead of oil. If you knead longer, it might taste a little bit different. So for our purposes, we need to know that there are 20 amino acids that our DNA codes for. But the combination of these acids and their interaction can change things significantly. The relationship between DNA and chromosomes. So each chromosome, the cookbook, consists of a single DNA molecule that is tightly spooled around clusters of proteins called histones and shown as purple balls in this diagram below. One cell, if you unravel the DNA, each of the 46 chromosomes, you line them up end to end, it's about 2 meters. So it's wound up pretty darn tightly in there. All right. Okay. Depending on how large it is, each chromosome contains hundreds to thousands of genes along the DNA strand, just like a cookbook can contain hundreds of thousands of recipes. So the gene is the recipe, and what you actually make is the protein. Okay. Recipes are written using letters that are ordered in a certain sequence to form words. Genes are written in letters too. Those are the bases. They are ordered in a specific sequence to form genetic words. The genetic words are called codons. So the genetic language is actually much easier than our English language. In our English language, we have 26 letters, and we put those in different combinations to form different words. So I guess the longest word in the English dictionary is here. It's got 45 letters. You can work on pronouncing it. I always thought it was supercalifragilisticexpialidocious, but I guess not. So there are tons and tons of words. But in the genetic code, there's only 4 letters, A, T, G, and C. And the words can only be 3 letters. So there's really only 64 words. Makes it a lot easier. So all of the chromosomes in a given cell are like a set of cookbooks in a library, and this is called a genome. And the goal of the Human Genome Project, which was completed in 2002, was to sequence all of the DNA on the 46 chromosomes in the human gene. All right, what do we need to know? The structure of DNA allows it to contain coded chemical instructions called genes. A gene provides the information for making a specific protein. There's 2 main steps from gene to protein, and mutations can change the meaning